Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Navigating IDE and IVDR Requirements in Drug Development. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank BioAgilytix, who developed the content for this presentation. BioAgilytix is the trusted partner for bioanalytical services throughout every phase of the drug development process. As a leading global bioanalytical laboratory, the company provides established and emerging pharmaceutical and biotechnology organizations with pharmacokinetic, immunogenicity, biomarker, and CMC analytical testing services in a GXP environment. BioAgilytix maintains state-of-the-art laboratories in Durham, North Carolina, Boston, Massachusetts, San Diego, California, Melbourne and Brisbane, Australia, and Hamburg, Germany. I'd now like to take a moment and introduce the speakers joining us from BioAgilytix today. We have today Marco Kling, PhD, Associate Director of Bioanalytical Operations, Luke Armstrong, PhD, also Associate Director and Bioanalytical Project Manager, Natalie Smith, PhD, the CLIA Laboratory Director, Amanda Hayes, PhD, a Scientific Officer, and Rob Nelson, PhD, Scientific Officer, and also Will Williams, MS, Senior Director of Lab Operations. But without further ado, I'd like to go ahead and hand the presentation over to our moderators for today's event. Amanda Hayes, you may begin when you're ready.
Thank you, Ryan. Thank you for the introduction and thank you for the opportunity for us to present today. I'm delighted to introduce this topic, and once I'm done with this brief intro, I'm going to hand it off to my colleagues, Luke and Marco. Next slide, please. All right, let's take a look at our agenda for today. So I'm going to introduce the topic of adapting to an evolving regulatory landscape with respect to IDE and IVDR. My colleagues Luke and Marco are going to give a more detailed overview of both regulations, and they're also going to present some case studies based on our experience. And finally, we're going to close this webinar with a panel discussion where I'm going to be joined by additional BioAgilytix colleagues and subject matter experts. Next slide, please. So at BioAgilytix, we have a very unique perspective on these evolving regulations and requirements. We've seen an increased number in requests for IDE in the US, as well as IVDR support in Europe. And it's well known that these evolving regulatory requirements have been a challenge, very challenging to navigate, and they've presented a lot of uncertainty in the drug development industry. And so today we're going to discuss on our experience with IDE and IVDR and examples that we have associated mainly with AAV-based gene therapies. So given our vast experience with developing and validating immunogenicity assays, we've been able to lean on that experience with some modifications to make these assays screening assays. So they're used typically for purposes of inclusion and exclusion from clinical trials, which subjects these assays to these IDE and IVDR regulatory standards. And our lab locations have really allowed us to be proactive in this space, given our extensive immunogenicity experience, as well as our established CLIA experience in the US, and our geographical footprint in Europe, in Hamburg, Germany, has allowed us to be active and spearhead this area of support for our assays. So next slide, please. All right, let's take a look at our strategies for IDE in the US. Essentially, for a medical device, a study undergoes a significant risk determination. This considers the assay either non-significant risk or significant risk, which then it's classified into a designated class. The strategies pursued here could require, if deemed significant risk could require a full CLSI validation prior to that IDE submission, or an option could be submitting an IDE application using a GCP or GCLP or a CLIA assay and awaiting for the FDA's CDRH response, either for approval, or there could be a conditional approval with additional validation requirements, or it could be conditional approval for a full CLSI validation. Or lastly, another option could be to remove the assay entirely from enrollment criteria. This would remove it off of that IDE path. Also going to talk about IVDR, quick overview. It tends to be a bit more complex because of the member nation specific regulations, in addition to general EU regulations. In our experience, Germany seems to be the strictest jurisdiction, which plays in our favor with our lab location in Hamburg. In the case studies today, we'll review IVDR requirements, following mainly the in-house exemption pathway that allows the use of a non-CE marked assay. And from here, after that overview with IDE and IVDR, I'm going to hand it off to my colleague, Luke Armstrong, and he's going to be discussing an overview of IDE pathway first.
Thank you, Amanda, for setting the stage. I'm going to kick off by describing the platforms that we are using for detecting antibodies against AAVs for patient enrollment. Next slide, please. So the assay platforms that we use are cell-based neutralizing antibody assays, NAB for short, also called transduction inhibition assays, and also plate-based immunoassays for total antibodies against AAVs, and those are TAB assays for short. And the diagram for the cell-based NAB assays is shown on the top right. And in these assays, there is a capsid of interest that carries a transgene for a reporter gene, usually luciferase, which generates a luminescent signal after being incubated with the cells. And pre-incubation of the capsid with samples, if antibodies are present against the capsid, they block the transduction of the cells, and that results in a reduced signal. And the diagram for the plate-based immunoassays is shown on the lower right. And in these assays, an unlabeled capsid is coated onto a plate. The sample is incubated, and antibodies that may be in the sample against AAVs will bind to the capsid. And then the contents are washed away that are unbound, and then bound antibody is detected with a labeled version of the capsid. So immunogenicity assays are conventionally used for evaluating treatment emergent responses, and AAV antibody assays are not an exception from that. In that case, for those purposes, they're validated under FDA immunogenicity guidance from January 2019 to satisfy GCP, GCLP immunogenicity reporting regulations. And in this case, an IDE is not needed as no treatment decisions are made based on the results. However, there is a concern in the fields that pre-existing antibodies against capsids may reduce efficacy of AAV gene therapies, and that patients should be tested for acceptable levels of antibodies prior to enrollment. And when these anti-capsid NAB or TAB assays are used for patient enrollment in clinical trials, clinical diagnostic regulations apply, which in the US means CLIA regulations. We find the assays validated for GCP, GCLP purposes are largely sufficient for CLIA purposes as well. There is an additional requirement for limited long-term stability for CLIA. And also when the assay is in production mode, actively being used for testing patient samples, proficiency testing and competency testing are required to maintain CLIA compliance. So that model was sufficient in years past, but recently these assays have been deemed by CBER, Center for Biologics Evaluation and Research, to be a significant risk to patient health and patient safety. And significant risk assays may need to receive an investigational device exemption, and that is granted by the CDRH, another arm of the FDA, Center for Devices and Radiological Health. And validation for IDE needs to be performed according to CLSI guidances. And I have a couple slides showing differences between the immunogenicity and CLSI guidances. And finally, development and validation for IDE purposes needs to be performed under design control. So next slide, please. So this slide depicts the design control process, and for scientists who are accustomed to developing and validating clinical trial assays for GCP, GCLP purposes, the design control paradigm might seem daunting. Some of that is just vocabulary. Design control doesn't really change the course of development and validation. It just requires formal documentation at each stage. And each stage has different nomenclature. So the process starts with defining user needs, and the users can be all stakeholders. That's primarily the sponsor for the trial and the assay, and also clinicians who would be using test data to enroll the subjects. There are also regulatory needs and also considerations for safety of the operator in the assay, for example. And at that point, once the user needs are agreed upon, the design inputs progress from that, and those are more detailed considerations of method, materials, assay procedure, the performance parameters to be tested, and the experimental design for those parameters, and also then specific acceptance criteria. And the next step, design process, that's the execution of experimental work. And design outputs are the results of the experimental work. And in the initial phases, that's the data coming out of the experiments. And this is an iterative process as development and optimization proceed. The assay conditions, as if they may change, are verified, and we go back to reconsidering the design inputs. But at the final iteration, when the assay has achieved the acceptance criteria for all of the design inputs, then the design outputs then represents the reagents and method SOP and worksheet that define the assay now/device. And at this point, the design of the assay is locked down for validation. Any changes would require a change control. And validation is performed to formally evaluate the assay/device in its final state for the parameters that are essential for reliable performance with patient clinical samples. And each step undergoes a review. It's informal during the iterative process, but there are formal design reviews at key stages. The design reviews occur at the user needs and design input stage, covering the development plans, and then after the final design outputs are achieved, but prior to validation, another design review occurs. After the validation is complete, there's another review of the suitability of the validation results, and then finally a review to monitor transfer to a manufacturing unit for proceeding into production. Another key requirement is that formal risk assessment is performed and that risks are reduced to be as low as possible. And all of this documentation is compiled into a design history file, and we've found that an IDE application does not necessarily include the DHF, but it may be requested at any time. Next slide, please. So this slide considers the components of the assay and how they may differ between the regulatory standards. First, in both regulatory standards, the matrix needs to be as close as possible to the intended test samples. AAV therapeutics typically target rare diseases, which may not have sufficient material to support validation. And so there is reasonable flexibility for use of normal individuals for the validation. A big difference exists with the surrogate positive control. For the immunogenicity guidance, an SPC with a defined concentration is needed so that a mass per volume sensitivity value can be assigned, and that usually means an animal-derived, purified monoclonal or polyclonal antibody. However, for obtaining an IDE, the CDRH has been very firm that validation parameters need to be tested using fully human samples and not with antibody or not with animal-derived samples. But there is no requirement that the samples have a mass per volume concentration. And then finally, for critical reagents for immunogenicity, lot-to-lot variability, of course, needs to be considered, but it's not really a formal validation requirement. However, for an IDE, the CDRH does want to see multiple lots used in the validation and lot-to-lot variability included in the precision table. And also, the CDRH wants to know the procedure that is intended to be used for qualifying new lots of critical reagents, and particular scrutiny is placed upon the capsid, both the labeled and unlabeled versions. Other considerations are for labeling and packaging the critical reagents, and also documentation and execution of the manufacturing process, which needs to be compliant with ISO 13485. Next slide, please. And this slide covers the validation parameters and illustrates how they vary between regulations. One requirement with such a distinction is specificity by analyte depletion. In the immunogenicity validation, there is a confirmatory step for TAB assays, which addresses this by competition, but NAB assays typically don't include a confirmatory step, but it's pretty consistent feedback from the CDRH that immunodepletion via protein AGL beads or AAV coupled beads is needed to demonstrate that the positive samples are indeed positive due to antibodies, and negative samples are lacking any antibody activity. Precision has some distinctions. Already told you about the need for human samples for IDE validation of precision. And for the devices category, the CV criteria can be more stringent. Typically 15% CVs are needed, with maybe some small accommodations for in some cases. Precision testing needs to be more extensive in terms of the design and number of determinations. And another key factor is the dilution at which the precision is assessed. Under immunogenicity, precision is evaluated at the minimum required dilution. But for an IDE, precision is evaluated at the clinical cutoff, and that cutoff may be a dilution that is higher than the MRD. And for a device, a low positive sample needs to be tested. That's pretty much the same as an LPC under immunogenicity, but one also needs to include a high negative sample with a low level of antibody that is not quite sufficient to render the sample positive. As far as sensitivity, I've already alluded to this parameter in discussing the SPC, and that's one of the few elements that's less stringent for devices. Stability is another important consideration. For immunogenicity, just samples are considered for stability. But for devices, critical reagents are also considered for stability. More of a concern for getting pre-market approval for a CDX, but it may be needed for evaluation for IDE. And for immunogenicity, typically just a couple of freeze-thaw cycle numbers and time points are needed for freeze-thaw stability and short-term stability. Device regulations may require a series of freeze-thaw numbers and time points so that drift can be assessed. And then finally, device regulations may require transportation stability to demonstrate samples are stable during the ordinary shipment process. Interfering substances is a very important parameter for device level assays. As typically for immunogenicity, we typically validate hemolysis and lipemia, but diagnostic devices require interference testing with more substances. Additional endogenous interference are needed. Rheumatoid factor, cholesterol, triglycerides are particularly needed, and also concomitant medications that are specific for the intended trial population need to be considered. So next slide, please. So in many cases, a GCP GCLP validation has already been performed when the call is made for needing to have an IDE, and we have had some success in obtaining IDEs by submitting GCP GCLP validations by adding on some additional validation to meet CDRH and CLSI requirements. And the key additional validation parameters include using fully human samples for all parameters, assessing those samples at the clinical cutoff, and also using the same plate map that would be used in sample analysis during the clinical trial. Immunodepletion needs to be performed to demonstrate sample positive, negative status, interfering substances with key additional endogenous components, and co-medications for the trial indication, and also a demonstration of lot-to-lot consistency of AAV capsid, both unlabeled and labeled, are needed. Next slide, please. And another scenario is a case in which BioAgilytix has an off-the-shelf assay that's offered to multiple clients, and one sponsor has already achieved an IDE with that assay, and then a second sponsor needs to obtain an IDE that's specific for their trial. And in such cases, a second IDE can be submitted that references the first IDE to the FDA, in such a way that the sponsor number 2 is blinded to the content of the first IDE, and that allows for much of the work that's been validation work that's been performed for the first IDE not to need to be repeated. But there may be additional validation, partial validation, to address any differences in the user needs between sponsor 1 and sponsor 2. For example, if the cutoff for enrollment is different, precision may need to be done at sponsor number 2's cutoff with the plate map to be used for sponsor number 2 if that plate map will be different. Also, interfering substances is a key one. The common substances between trial populations 1 and 2 may not need to be retested. Caveat there is if the cutoff is lower in trial number 2, then there's probably more need for interfering substances reevaluation. And of course, any co-meds that are unique to the number 2 trial population would need to be tested. And also, lot-to-lot variation of AAV capsids is a big concern for the agency. So demonstration of comparable results between lots used in IDE 1 versus IDE number 2 is important. And if lots for IDE 1 are not available, then at least a lot-to-lot demonstration of consistency would be needed if that hasn't already been provided. So my time is up, so I will yield the floor to Marco to tell you about how AAV antibody assays are managed in the EU as investigational devices. Thank you.
Yes, thank you, Luke, for setting the stage on the IDE part. And as you correctly mentioned, now we are switching over to the EU, where you will find some parallels, but also some differences when looking at IVDR requirements, especially when focusing on the in-house test exemption. Next slide, please. So here we have an overview of different pathways, how you can comply to IVDR. The easiest one, if that's available, which is not the case in most of the scenarios we are working with, is the use of an already CE IVD marked test on the market that exactly fulfilled the intended use of the study you might be working with. Of course, that would be an opportunity to have an easy access, a direct access to initiate quite quick testing. But most of the time, the CE kits that are available are either not fitting the intended use, or they are just not existing, especially in the field of gene therapy. In the history, there have not been any CE mark tests for the detection of anti-AAV antibodies. So in most of these scenarios, we need to find other ways to comply with IVDR. Still the gold standard pathway, also the way to CE marking, if you already have an assay in place and you've built an assay under design control that fulfills most of the analytical criteria, but still lacks the performance data, would be running a performance study for the device. So this is an opportunity if there has been substantial work already done, also for example, in a non-EU lab, but it comes with some challenges as well, as an IVD performance study is not really a small undertaking. It's a quite complex setup, very similar to a clinical trial. You need to get green light in each and every member state in Europe that you want to apply for. So it's quite a huge effort to take. The approach we are focusing mainly on at BioAgilytix is the in-house IVD. And in-house IVD is an IVD-like test with a full diagnostic purpose, but without a CE mark. It comes with some exemptions, but also with some requirements. The requirements being that the lab that is manufacturing that test is a single health institution in the EU and has an appropriate quality management system in place and complies with ISO 15189, and there's no distribution to third parties. So the test is purely manufactured and used in the same facility. Especially for early clinical trials, this provides, in some cases, a quicker access to start testing already in phase 1. Before you need to put a lot of effort in running a performance study. So it could bridge that gap also later on for bridging the time until a CE mark may be granted from the EMA. But also there, of course, there are some challenges on the single site for the testing. And if you have already an existing assay in place outside of the EU, there may be a requirement to transfer it in. I want to focus a bit on a case study on the IAV space we have worked on in the past. And if you jump to the next slide. So this is a typical scenario we are facing in the discussions with our clients, that there is a clinical phase 1 study where the scope of the project is to assess neutralizing antibodies to an AAV capsid. Most cases, as already mentioned by Luke, we have validated these assays for GCP requirements, but now the clinical trial protocol is putting the assay in scope of IVDR by including an inclusion-exclusion step up front to only include patients that don't have pre-existing antibodies. So you see the scheme that Luke also showed. I will not go over this for the sake of time. But then the question comes up, how to address the regulatory requirements for IVDR for such a test. If a GCP assay is already available, of course, it doesn't change the need to be able to detect the neutralizing antibodies against the capsid. According to the IVDR, we don't need to only demonstrate analytical performance, but also clinical performance. The validation, should follow the guidelines for diagnostic assays, but also at the same time as all enrolled patients will still be measured after dosing. It must also fulfill the current guidances for EMA and FDA. So you can see this as a hybrid. Or from the other perspective, what is actually really different between the full-blown CLSI guidelines and the GCP validation? What can be done the same way? And is it always really required to go the full-blown way, especially in early clinical trials, as the IVDR is also focusing on using state-of-the-art methods for the specific fields of interest. So this is why I started to deal with that strategy. And if you go to the next slide, you saw a similar slide in Luke's presentation. You could go through the parameters that are used in the GCP validation, like cut point, sensitivity, selectivity, specificity, and so on. And you can look at the CLSI guidelines, what is actually the definition of the parameters they are using there, like limit of blank, limit of detection, interfering substances, and so on. And if you do this, and I've put in some references here, is that, for example, for the cut point, if you look at how CLSI is defining the limit of blank, this is more or less the same approach we're using to set the cut point. And this is the way how we were dealing with the scenario in this specific case study, that we were kind of intertranslating the terminology of both worlds without really changing too much on the experimental setup to fulfill the requirements of both guidelines and to facilitate it for the reviewers that are specialized on the respective guidelines, that they find their terminologies back. Next slide, please. So if you set this as the scene, you could think of different strategies how to kind of upgrade the GCP assay that you already have in place. The first consideration, which was also already mentioned by Luke, plays the same role in the world of IVDR, is that most GCP assays typically use animal-derived surrogate positive controls. We have heard feedback from FDA, but also in the meantime from EMA, that purely relying on animal-derived material is not the best scope for demonstrating clinical performance in a diagnostic assay. So it would be good to also use true human positive controls, for example, using samples with confirmed titers or antibodies against the capsid. But the question is, do you need to decide which one to take? Here we decided to use also a hybrid approach, to use human positive controls, especially for the critical experiments, like demonstrating the performance, the clinical sensitivity, so to say, that we're able to detect antibodies in real human samples. But on the other side, also use a surrogate positive control to understand the capability of a technical sensitivity, how sensitive is that assay, and also on the other hand to fulfill both of the guidelines when this assay can then also be used after the screening period for the subjects that have been enrolled into the study. Next slide, please. So another big difference, and you've heard this from Luke, is if you strictly follow the CLSI guidelines, which is the common expectation for IVD tests, would be a quite tremendous precision study, where I've put this on the bottom left. For each parameter or for each precision study you're running, you would be running 2 days, 2 runs per day, 2 replicates per runs, which would end up in 80 replicates per sample. And if you want to replicate this for multiple instruments, multiple lots, and so on, which is the recommendation, especially for commercially used assays that are distributed around the world, then you can end up with a quite tremendous number of replicates. On the other hand, if you look at the precision requirements in the GCP world, it is acceptable to have 6 inter-assays in a non-balanced design, 3 days, 2 operators, 2 replicates per run. So you're ending up with 12 replicates. And we were asking ourselves, how are we handling this? Do we go the CLSI way? Do we go the GCP way? Or will we pick something in between? And if you jump to the next slide, this is what we decided to go with a hybrid solution with 3 samples that are tested in a non-balanced approach on 10 days, using 2 operators per day, 2 lots of cells, and 2 replicates per run. So at least we were ending up with 120 replicates per sample. And in total, in a non-balanced design, 4 operators and 3 instruments were involved. You can see the precision data highlighted in both the overall CVs, which all met the acceptance criteria. And then if you put, yes, thank you, at the data from the GCP assay that had been previously validated, you can see that the CVs are slightly better, but there's no significant difference on the performance of these 2 assays. Both assays were absolutely meeting acceptance criteria, and there's actually no real benefit of adding more replicates, and this was demonstrating that the GCP assay is probably more than sufficient, especially for the early phases, and when considering that the test will only be used in a single laboratory, and there may not be a need to follow the CLSI guidance also on the percent CV expectations of 15%, as this assay is totally fine regarding its performance. Next slide, please. So this is on the lab work end. Of course, as you may imagine, even if we build an IVD, an in-house IVD, this doesn't mean we don't need additional documentation, because the requirement, according to Annex 2 of IVDR, is required, which is probably the most laborious part to get in place. So this is all about the device description and specification, labeling, design manufacturing information, the general safety and performance requirements. One big package is the benefit risk analysis and risk management. And just at the very right, the product verification and validator, which is just purely the little part that I just showed before on the lab work. So that's a huge effort to take. And even if we are exempted in the in-house exemption from full design control, there's still a need to provide a lot of documentation to support diagnostic testing. Next slide, please. The second strategy may be in the scenario we're also facing quite frequently, that the assay has already been established in the US following the IDE approach. This means that in these cases, a lot of data is already robustly established following design control. So they may, if we want to go that strategy, there may be significantly lower validation or verification work to be done. We may dramatically reduce the scope of work by not resetting the cut point, but just verifying cut point sensitivity and precision. If stability has been demonstrated, there wouldn't be a need to repeat. Also, the comprehensive technical documentation would be already in place. So there could be a lot of work that could be taken over from that assay already. But one of the showstoppers frequently is the timeline, as often the screening is supposed to start simultaneously in Europe and the USA. So this makes it very challenging to rely on the finalization of one package to then only start the transfer to Europe. And then one point which you learned from Luke is the intended use of the IDE, which may be different, or in that case would be different from the intended use in Europe, as the IDE is always study specific, while the in-house test is study independent. And I put a little example on the next slide. And if we jump over there. So on the left side, you see the intended use of the IDE test, which could be the qualitative detection of NAPs against that AAV capsid neutral serum samples from adults to support the exclusion of NAP positive disease X patients from clinical trial, whatever. The problem is that we cannot just take over that intended use for the IVDR test, as at this stage there is no performance data to support that these claims can be made, as there has not been any patients to support that. So the intended use in the phase 1 of the in-house test would be a bit simpler, just stating qualitative detection of neutralizing anti-AAV antibodies in urine serum samples from adults. So that removes the clinical claims from the disease indication and purely really relies on positive and negative results in urine matrix samples. So this test can then be used for inclusion-exclusion purposes in phase 1. And as soon as you collected your data from the phase 1 and you have reliable, robust clinical data with real clinical samples, the sweet thing about the in-house approach is that we can, based on the new clinical performance data, adjust the intended use of the test to more and more tailor it to the specific compound that we are working with, and to guide this assay through the following phases. So this was just a little overview of how we approached IVDR needs in one of our case studies. And now I'm handing over to Rob for a conclusion on today's webinar. Thank you.
Thank you, Marco, and thank you, Luke, for the IDE presentation. So yes, I'll wrap up the key points, key takeaways from today's webinar before we move into the question and answer session. So as I heard today, the regulation of non-commercial assays used for clinical trial enrollment has increased, and many of these assays are now reclassified as investigational devices. We've seen case studies today where the use of immunogenicity assays, whether that be total antibody TAB or neutralizing antibody NAB assays for patient selection, may trigger the need for IDE or IVDR compliance, depending whether in the US or Europe, and potentially later the need for companion diagnostic. We are supporting sponsors who are currently midstream in their clinical development, and we've supported them to retroactively comply with these new requirements. We've seen that assays that are originally validated for use in clinical trials provide a very strong basis and can be leveraged for achieving investigational device status and complying with the IVDR requirements. But there are some additional add-ons in validation, some requirements there. Achieving this device status often involves further validation, and we've seen from both Luke and Marco, the use of fully human samples aligned with the clinical assay cutoff is often a need. Testing of interference substances is much broader within the IDE and IVDR space than in the clinical trial GCP assays. We need to use the same plate maps we're going to use for analyzing samples with the device. And the validations typically follow the CLSI guidelines, although there are similarities across with what we do in the GCP world. And we've seen also that the development, validation and manufacturing of assays that regard devices need to follow the design control principles. So we'll wrap up there. I'm going to hand to Amanda, who is going to just give a couple of slides presentation on who we are at BioAgilytix before we go into the Q&A. So Amanda.
Thank you, Rob. Just real quickly, before we get into the panel discussion, for audience awareness, if you're not familiar with BioAgilytix, we're a global bioanalytical service company with labs in the US, European Union, and Australia. Next slide, please. And we provide assay development, validation, and sample testing for bioanalysis for every modality and every phase of drug development. So pharmacokinetics, immunogenicity, biomarkers, and CMC analytical. And with that, I'd like to invite our panelists for a Q&A session.
Great. So we see questions coming in already. So please do use the questions box and put your questions in there. We'll make a start with what I see here. So Luke, Natalie, we've heard discussion today about the IDEs and kind of like a single lab test and device. And yeah, I was wondering, one of the questions is what's the difference between kind of IDE, LDT, or similarities, and how do these latest political changes in the US impact the IDEs with the LDT final rule change?
Yes, I'm happy to start it off. Yes, so the FDA's final rule regarding LDTs. So when that originally came out earlier last year, essentially we have two different branches of the Department of Health, the FDA and CMS, the Centers for Medicare and Medicaid Services. And up until this point, LDTs or lab developed tests had always been regulated by CMS, but the FDA was looking for more oversight and more stringency in how these assays are validated and overseen. So they proposed a between 3 and 4 year rollout of medical device manufacturing requirements. And so, but after a recent court ruling out of a Texas court, it was actually vacated, and so it's no longer applicable. Even when it was currently in play, with assays inside of a clinical trial, they're not commercial assays, so it wouldn't apply at that point. When an assay is deemed significant risk, you have you comply with IDE requirements, and if it's non-significant risk, the abbreviated IDE requirements. But then it's really in the commercial phase, when you move into a companion diagnostic, if it's needed, you would already be required to comply with those medical device requirements. So either way, we were, covered on either side. But I hope that gives a little bit of a, basically, it doesn't change anything for how we're working here. We're still complying with IDE or IVD, well, IDE requirements for the US, and then we would still be complying with companion diagnostic for any commercial bids.
Thanks, Natalie. And then we have a question that I'll direct to Will and Marco. So we must have cases, or I know we do have cases where we have an IDE in the US and IVDR in Europe for the same assay. What kind of requirements are there, or what are we doing to kind of cross-validate or to, I guess, assess performance of those assays that are running across the different sites?
That's a great question. It is something we've done, like you said, Rob. I think at minimum, we want to understand and make sure that there's two assays performing on two different continents, that they're performing to what degree they are similar. We want to, if we can, make sure we can make those as similar as possible. As Marco alluded to, and I'll maybe turn it over to him too some, is that, depending on the use case of the IVDR versus the IDE, there, how far we take that cross-validation and how, involved it is may be based on how the use cases for the two study wings of the study may differ. But we would look at that, and it would often involve looking at potentially patient sera, as we've alluded to. Human donors are critical, and understanding how those perform is one of the key things we would look into. But it's certainly something we would advise most people to at least consider.
Yeah, I agree, Will. I think especially on a few parameters, if there has been a full establishment of an IDE assay, as mentioned already in the presentation, I would not revalidate everything, but make sure we have the same performance in a verification study, checking, for example, the positivity rate, checking the cut point, checking the sensitivity of the assay, to really ensure we get the same readout on both continents, which can be crucial, especially if it's a global program that's running. And yeah, I think that's the best way to approach it without investing a full validation on the other side.
Maybe to talk a little bit more about IVDR, this is a question that we get a lot and have a lot of discussions about, and understanding what's applicable, what's in scope for IVDR. How do we handle those conversations? Is there, you know, a black and white, in scope, not in scope, and how do we navigate that? And this question's for Marco.
Yeah, thanks, Amanda. I think sometimes it's not really easy to define this black and white, because it depends on a lot of factors, and mainly exactly how the protocol is written. So I think this is an important message, that by writing your clinical trial protocol, you can maybe also unintentionally put something in scope which you actually don't intend to be in scope. And so what we try in the discussions is to really understand the need for the diagnostic readout. Is inclusion, exclusion really required, for example, and to challenge the sponsor with our questions, and then to find the best solution moving forward. Because in some cases, there's probably no need to put it in scope. We had an example discussion on replication competent lentivirus testing, which is a crucial assay in the CAR-T world. Is that in scope, or is that not in scope of IVDR? Because you are kind of monitoring the patients for RCL, and you would stop the study once you end up with a positive patient. And there was a really controversial discussion at one of the IVDR meetings here in Europe, and I think still this is not really black or white, and you can find arguments for both ends, but in the end it really comes down to how are you writing your protocol, and what's the end point you're targeting.
Okay.
Go ahead, Rob.
Going to say, staying with Marco, you have a question on one of your slides around the hybrid validation, so kind of the hybrid GCP CLSI validation, and whether that's been approved or viewed by a notified body and accepted, that approach.
So it has not gone through a notified body, because that would be in the stage where we are trying to achieve a CE mark, and this is now phase 1, right? So we are very early in the clinical development, so there was no involvement of a notified body. The FDA was at least fine with the GCP-based validation, as we had set this up earlier, and since the diagnostic decision-making is more on the European end, the FDA was not involved in that discussion. So then also maybe there's a difference in the in-house approach in Europe. There is no pre-submission like for the IDE in the United States. So we are building that assay and filing all the technical documentation, and need to be able to provide it on request. And so far, or at least for this case, there was no specific request on that end.
Okay, so I see another question there. So we've mentioned that we're working on assays that fall under IDE and IVDR in Europe. How do we coordinate those when we've got testing on 2 continents in terms of data reconciliation or working with the sponsors across those sites? So maybe Luke and Marco again.
Luke, you want to start?
Yeah, so actually there's CLSI guidance on that as well. Concordant studies with a panel of samples with a range of activity should be performed as those same samples being tested at the 2 different sites, and concordance of the results being determined, and statistical analysis to see if there's any bias occurring. Is the formal way to demonstrate equivalence across sites. Anything to add, Marco?
I think this is absolutely right. Besides the points I already mentioned when making sure during validation phase that the assays are performing equally, that's definitely an add-on that we are performing to make sure we have a defined set of samples that is performing equally on both continents. And still, of course, there are different requirements in the IDE and the IVDR world, and it's not always easy to tackle them both and to find a good alignment. So sometimes we need to have specific solutions for the US and the EU, but where the points where there's a lot of harmonization, like CLSI is a good starting point. In any case, we are trying to do that.
And I know we talked a lot about analytical validation and some of the differences there. We've discussed CLSI and maybe how we've adapted that to these assays. So maybe a question to Luke and Will here. After approval of an IDE, what happens if a cutoff is changed? Can you speak a little bit to what happens with that assay validation?
So the guidance that I've understood from the CDRH based on pre-submissions addressing this is that definitely precision needs to be reevaluated at the new cutoff. Other parameters may or may not need to be redone. So, you know, interfering substances, basically it probably doesn't need to be redone unless the dilution is lower, and you would have to retest the samples, and they would have a higher effective concentration of the interferent in the test.
And maybe to follow on to that with US and Europeans, are there differences in bringing up assays in Europe and the US in terms of how you approach that?
Maybe I don't get the core of the question, Rob.
Are most of the cases where you've dealt with where you've already got an existing assay, or are you bringing up assays from scratch? And would that change your approach?
So I think from the regulatory perspective, it doesn't really matter if you build the assay from scratch or if you have pre-existing data available you can refer to or in-source. That really depends on the specific program. In the end, how much work you need to perform. In the end, you need the full package available. I think this is obvious. And the question is, where does that package come from? Does it come from already existing data, or do you need to regenerate it from scratch at this point? But the overall approach is not different. We make an assessment, what's already there, what do we need, and then we go ahead. Anything from your end, Luke?
No, I think that covers it.
So I know we talked a lot about gene therapies and these immunogenicity assays for inclusion-exclusion. What other modalities would these regulations apply to? IVDR and IDE?
I mean, I think it's any assay that's being used for a result that is going to be used for determining enrollment in the trial. Yeah, so there's, I understand that there are some molecular assays, for example, that that necessitate that.
And yeah, also biomarker assays as well. So if you want to include or exclude based on a specific biomarker cutoff value, that could be in scope as well. We try to keep PK assays out of scope if possible, because Usually they're made for safety and efficacy testing. We know there are discussions of programs where PK is put in scope. There may be some justifications, but I would say 99% of PKs should be out of scope.
Okay, so we're at the top of the hour, so I think we will thank everyone for joining us for this discussion. If your question hasn't been answered, we will get back to you. And if you have any follow-up questions, I'm going to put the details up on the screen now, so please feel free to contact us. And with that, I'll hand back to Ryan.
Yes, thank you so much for this very insightful presentation. As you've heard, we will make sure that BioAgilytix will follow up with you for any of our questions that we couldn't get to. We've got some contact info on screen. In the chat box, I've sent a link where you can view the recording of today's event. And then I also encourage you, though, to share this with your colleagues so that they can register for the recording here as well. Also note that as you leave, a survey window will be popping up on your screen, and your participation is appreciated as it helps us to improve our webinars. Now, please join me once more in just thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you.
Thanks, everybody.
Thank you.